Well, the students in my class, we could ask a little question near the end of the class for them to answer to turn into their TAs. But one question I want you to start thinking about, and I'm hoping Jeff will say something about this. Of course, humans have been a kind of a natural selection factor. I guess we call it artificial selection when humans do it. But we have been shaping plants and animals that we harvest for food for a very long time. We have exerted human ingenuity through breeding to come up with particular characteristics in plants and animals. And I'm hoping you'll be thinking about, and Jeff will say something about, what's really different about this genetic engineering. It's also using human ingenuity to reshape plants and animals. How is it so different from what our ancestors have done for thousands of years as they've interacted with them? So I'm going to turn it over to Jeff now.
Thank you very much. Okay. All right. Is this? Are you going to be able to hear me? If anyone can hear me, raise your hand. If you can't hear me, raise your hand. All right. So then you can hear me. In the last 3 years, I've traveled to 25 countries and 6 continents, about 180 cities. I've spoken to hundreds of senior politicians and reached over 100 million people. This is the first talk on environmental effects of genetic engineering that I've ever given. What I talk about generally are the health effects and the compromised regulatory agencies, the industry manipulation, etc., that got these foods approved. And then during the questions and answers, people ask me about transfer of genes to soil bacteria, invasive species, contamination, non-target effects on monarch butterflies, and that's when I get to enjoy giving the realm of environmental impacts, which I have strategically left out of my talks. I have strategically left them out because I have a purpose for publishing books and creating DVDs and whatnot. It's because I believe that genetically engineered foods are extremely dangerous and should be removed, and I'm figuring, what do I need to say to convince people or to create a momentum to remove them from the market? And my personal experience, speaking to activists in all these countries and seeing what I've been seeing in the history of this dangerous technology over the last 10 years, is that consumers are on the top of the food chain as far as GMOs are concerned. And in Europe, where people know about it, concerned consumers forced rejection of the GM foods by the European food manufacturers. And so I'm trying to figure, well, what do I need to do to have that same kind of rejection in the United States? And I can talk all I want about the environmental impacts, and people will say, yes, GMOs are bad, yes, they can hurt agriculture, yes, they can hurt the environment. And then they'll go to the store and they'll buy the same products that they've been buying all along. Sort of a disconnect. Most people don't make purchasing decisions based on environmental impact. Maybe you do, but you would be in the minority. So strategically, I have chosen health effects because most people, when they realize how dangerous the foods are, they may choose not to risk eating them or eating them on a large scale. So I'm going to rely on you today for asking a lot of questions. I'm going to give the talk in a short way, and then have you ask questions so I can go deep into areas that I don't usually talk about but study all the time. But I'm going to answer the first question, what's so different about genetically engineered crops compared to the traditional breeding? With traditional breeding, you take... Pay no attention. You take male and female or parent crops and you cross them, parents, and the genes interact in an organized way, reshuffle in a certain way, and you end up with offspring that have some characteristics or genes from either parent. Genetic engineering is entirely different. For example, let's say you want to create a pesticide-producing corn. So you'd go to the Bt, Bacillus thuringiensis bacterium in the soil, and identify the gene that produces the Bt toxin, and you remove it from the DNA. Now, the bacteria genes code for proteins a little differently than the genes in plants. Certain amino acids have a different formula, so you change the amino acids a little bit in the bacterial gene, and you put it into the DNA of the corn. Actually, if you just did that, nothing would happen, because the DNA has a self-regulatory mechanism so that it turns on the genes that it needs to at the time. In the part of the plant that needs it, and during the season or the time of day that it needs it. It's a very intelligent system. We don't understand that language. So the developers want that pesticide to be produced in every cell 24/7 around the clock in high volume. So they take a genetic material, a promoter, whose job is to turn on genes. They take a promoter from a virus, which is very aggressive. It turns it on constantly. And they take that and they attach it to the gene. So it causes an overproduction of this Bt constantly. So it's never in the control of the DNA that it goes into. And they typically put an antibiotic-resistant marker gene in there as well. I'll tell you why in a minute. And then they make thousands of copies of this transgene, or gene cassette. And they typically put it into a gene gun. And then they blast it, thousands of genes, into thousands of cells, hoping that at least some of those genes make it into the DNA of some of those cells. They can't tell which ones get in there from a microscope, so they douse it all with an antibiotic, killing all of the genes, all of the cells, except the very few whose DNA incorporated the gene cassette, which included the antibiotic-resistant marker gene. And that's what caused it to survive the antibiotics. That's what tells the scientist that the gene cassette got into the DNA, and then they grow plants from those cells using tissue culture or cloning, and then they find which has the agronomic characteristics that they're looking for, and then they commercialize it. What can go wrong? This is what I'm about to describe are the things that can go wrong with process of genetic engineering, which make it very dangerous both for health and the environment. In the first case, the process of growing the cell from tissue culture, even before you insert the gene, creates mutations up and down the genome. Hundreds or thousands of mutations are possible. Now later, when you back-cross it with other species before you commercialize it, some of those mutations are eliminated, but not all. And for things that you don't back-cross, like potatoes, all of those mutations from the process of this tissue culture remain in the potato. So now you're introducing a crop with hundreds or thousands of mutations just from the tissue culture. Then you also blast it with a gene gun, one of the two major varieties in gene insertion, and that causes damage right around the place where it gets inserted. It can knock off genes. In fact, they found in one case 40,000 base pairs of DNA were transferred to a different chromosome. In one case, 13 genes were deleted. You can delete genes, you can turn them off, and fragments of the transgene can end up throughout the genome. You also have that promoter, right? The promoter which turns on the gene from the virus. It can accidentally turn on another gene downstream from the inserted gene, 24/7 around the clock, overproducing a potential toxin, an allergen, a carcinogen, an anti-nutrient, something that will cause vulnerability to disease, or something great, we don't know. So there's a lot of unknowns in the process of insertion. And then they introduce it into the croplands without actually evaluating all the changes in the DNA. Now, what does that mean for the environment? It can mean a lot of different things, because it really is a kind of a genetic roulette. And genetic expression these days is undergoing a revolution. In fact, every few years it goes under a revolution. And so this technology was based on obsolete science. Most of these key assumptions have been overturned since it was introduced. And I want to say at this point, I'm not against the possibility that someday in the future, engineers can safely and predictably manipulate the DNA of plants and for the benefit of health and the environment. But in the current generation, we're seeing a primitive technology based on an infant science where it's being released into the environment where it can never be recalled and fed to millions of people. So you have these potential changes. Now, what could go wrong? Well, the first time cotton was introduced in a big way, BT cotton, cotton that produces its own pesticide, or Roundup Ready cotton, cotton that you can spray Roundup herbicide and not kill it. I'll explain that in a minute. It was quite unusual because the bolls, the cotton bolls, were just falling off the plant. In some cases, the taproot, which normally gives the stability to the cotton, was unformed and the plants just fell over in the wind. These unintended effects from the process of genetic engineering cost farmers millions of dollars, and they settled with Monsanto, who developed these seeds. When genetically modified soy was introduced, in the hot sun, all of a sudden the stems started to split. And they found, when they checked it, there was a high lignin content, the woody element. And so what happens is we don't, under certain circumstances, with this, there's changes in the genome that we weren't aware of. And this revolution that I'm talking about is called epigenetics, where they're realizing that environmental factors and stress factors and input factors can change expression levels of the DNA in ways we were not aware. In fact, the food that a mother eats while pregnant can influence the gene expression of her offspring and her great-grandchildren. So this is a whole other understanding of DNA that we weren't aware of. We're understanding now that the same plant, grown under different environmental conditions, in different temperatures, can have completely different expression of proteins and secondary metabolites called natural products. So imagine if you've turned off a gene during the process of insertion, if you mutated 1,000 genes during the process of tissue culture, and you've only tested it under ideal circumstances. And now you put it into Montana 3 days ago, and it's 25 degrees below zero, where I was. And all of a sudden, the natural response of the genome that was occurring in corn or cotton or whatever is now distorted because of something that was never checked, because it was never field tested in 25 degrees below zero. Just like the high lignin content, just like the drop-in bolls. And so what we see now is a series of unintended side effects that are occurring over and over again with genetically engineered crops. Another possible cause of problems. See, that's unpredicted side effects because of collateral damage in the DNA due to the process of creating the crop. Another unique problem is that you're introducing a trait that has never before been part of that species. You now have corn that produces its own pesticide. And you can think in terms of drought resistant or salt resistant. So you're all of a sudden changing very significant agronomic traits. Right now there's only two main traits. There's creating its own pesticide, or there's resisting death by herbicide. Now, many people believe that the transfer of these traits across the species barrier can create, it's kind of reductionist thinking, like you can add a gene like a Lego and just add this one trait and it's not going to cause a problem. When you look at environmental studies, you realize that that same reductionist thinking, as opposed to systems thinking, has been responsible for huge environmental catastrophes. Well, the entire genetic engineering of crops is based on this reductionist thinking. Let's just add a trait. So, let's look at some of the health or environmental consequences of the traits that are already out there. For the BT, it kills insects. It kills insects with an alkaline gut. It causes ulcerations in the stomach and it kills the insects. However, there are beneficial insects that eat the pest insects. And in one study, the beneficial insect that ate the pest insect had a damaged mortality rate. So now the Bt toxin, just like DDT, is being passed up the food chain. Also, other non-target insects end up dying in the presence of Bt. The most famous was the study in Cornell of the monarch butterfly, where they just put some pollen in the laboratory, some Bt pollen, which creates this pesticide, and there was more than a 50% mortality rate among the larvae, which was supposed to grow into monarch butterflies. And that is, it's questionable whether in actual field studies the monarch butterflies are at risk, although I heard recently there was a 75% reduction in their population, which may have been contributed in part by the presence of Bt, or possibly industrialized agriculture, which is eliminating the milkweed plant, which is where they deposit their larvae. And you also have the Bt, which is created in every cell of the plant, including the root system. Now, Bt is a pesticide that's used in organic agriculture. And they spray it in times of high infestation only. And then it biodegrades in about 2 weeks, most of it within the first 24 to 48 hours. And when you eat the product, it can wash off. The Bt in the corn and in the cotton is produced in every cell 24/7. So, and it's produced in the roots. So it's getting into the root supply. It's getting into the microorganisms in the soil. And it doesn't degrade there. In fact, it binds with clay and it has been found there as long as it's been tested. The first time was 8 months, then it's been found 2 years. I don't know what the latest is, but it's there on a stable basis, and it can alter the very important ecosystem within the soil. So we're talking about Bt in very high concentrations that can damage crops, or damage the pest life and the beneficial predators, as well as the non-target insects and the soil organisms. In addition, when you put out there a pesticide year after year in chemical form, traditionally the pests adapt through epigenetics and genetics and selection. They become immune more and more to a particular herbicide, or pesticide, excuse me. So before, Bt was used very sparingly by organic farmers as needed. Now it's out there in millions upon millions of acres in high concentration, causing pests to adapt to it. So what does it mean? It means that Bt, which is a very important herbicide for use in organic agriculture, will become obsolete much more quickly. And when you ask Monsanto what they think about it, who's the big biotech company that puts out a lot of this, they say, oh, don't worry, you should see what we have in the pipeline of other pesticides. Not necessarily organic, not necessarily as safe as Bt. In addition to Bt, the other major category is herbicide tolerance. Monsanto found a bacteria growing in a dump site near their herbicide factory, and it wasn't being killed in the presence of the herbicide, Roundup. So they figured, let's put this in the food supply. They took the gene out which produced the enzyme which broke down the Roundup and put it into soybeans. And so now you can spray soybeans with Roundup herbicide, and it kills all of the biodiversity around in the field, but the soybeans survive. So if you're in my state and you go around and you look at a soy field, you can tell if it's Roundup ready because it's completely clean. Clean is an industrialized concept. Sick would be more of the concept in terms of biodiversity. A dead zone, in a sense. And so when you eliminate that biodiversity, you eliminate the microcosm, the environment there, and the birds are affected, and the bees are affected. In fact, there was a field-scale trials done in the UK, where they compared herbicide-tolerant crops compared to conventional crops in sugar beets, canola, and corn. And in the canola and sugar beets, they found that there was a huge change in the weed characteristics around and a drop in the number of insects and a drop. They predicted that if this were planted in a wide scale, certain bird varieties would actually become extinct because they wouldn't be, they are the top of the, or they're part of the food chain which was being eroded by the presence of these herbicides that were wiping out the biodiversity. The corn, however, was worse with the conventional side, but they were using atrazine, which is so dangerous, it was actually banned before the field trial ended in Europe. It's not banned in the United States, but it was banned in Europe. So you end up with a reduction of biodiversity because of the spraying. You also end up with herbicide-tolerant crops, just like the pesticide-resistant pests. You have herbicide-tolerant crops. So what's happening now is there's pigweed and horseweed and a number of other very serious weeds which are no longer treatable by Roundup herbicide because it's used in such high concentrations. In the first 9 years of genetically modified crops, there was a 138 million pound increase in the use of herbicides because of those crops. So as an example, by 2004, it was estimated that there would be approximately 85% more use of Roundup on a genetically modified soy field than a non-GM soy field. So you're drenching the soil, you're drenching the biodiversity with this Roundup, you're creating Roundup-resistant, or glyphosate is the active ingredient, resistant herbicides, resistant weeds. So it's causing a shift in the weed population, causing farmers to use more toxic herbicides to kill those weeds. And so paraquat and 2,4-D and these other very serious, dangerous stuff are being used in part because of these herbicide-tolerant crops. Now, the herbicide-tolerant crops also lend themselves to industrial agriculture because you can spray with planes and just wipe out the biodiversity of an entire region and pump out these soybeans and make a lot of money. And that's what they're doing in Argentina, where they realized that they make more money per acre by turning this into large industrialized Roundup-ready soybean fields. And so they're cutting down forests, they're cutting down rainforests, they're kicking out the indigenous workers who've been working fields for years and generations. 91% of the population now live in the cities. 500 villages were abandoned. More than 50% of the population lives below the poverty line, and you have this ocean of Roundup-ready soybeans in Argentina. And you have planes that are spraying Roundup herbicide combined with other herbicides, because Roundup is not completely effective there anymore on its own, and it actually gets oversprayed on some of the peasant farmers in the area, causing sickness and death to them and their livestock, and death to their gardens and their farms. I was reading yesterday about an 11-year-old boy that got oversprayed in South America and died, and the food he was carrying home to his family also got poisoned because of the herbicide, and the 22 members of his family and friends that ate that also got sick. And the people who were in charge of that are going to jail for 2 years. So we have a situation where we have a monocrop. Now, it's unprecedented in Argentina. It's spreading into parts of Brazil, it's spreading to parts of Paraguay, and it's changing the whole landscape. And there's now, with that monocrop, you end up with some of the features of industrial agriculture. There's something called soybean rust, which is a very dangerous infestation. And you have the problems of the biodiversity. A friend of mine was down there, a filmmaker, was down there doing a study on Argentina, and she said it's just devastating. It's just a dead zone. When you go there, there's no insects, there's no weeds, it's just dead. So talk about environmental impact. The biotech industry considers it a great success. Now, Roundup was always considered to be a fairly benign herbicide. However, studies showed that in very low concentrations when put into water ponds, it caused a complete loss of certain tadpole species and a catastrophic decline of others. So it has a huge impact on amphibians. It was also expected to break down quickly in the soil. However, Denmark found that it had actually penetrated the soil into the water table and was present at 5 times what was considered to be safe for drinking. So they banned it in Denmark. So we now have drenched water supply, drenched fields with a potentially toxic herbicide. And just as a footnote, most of the tests on herbicide just test the active ingredient. There are dozens of, quote, inert ingredients in herbicide which actually are active themselves and work in concert with the active ingredient, which make Roundup, for example, much more toxic to human placental cells, much more toxic to cell development, much more toxic in general. One of the things is it has a surfactant which causes it to penetrate into the cells. That same surfactant causes it to penetrate into the skin. And yet that's not tested for in most of the safety studies. So you have the two main crop varieties, herbicide tolerance and Bt. There's a third, which is used in Hawaiian papaya, zucchini, and crookneck squash. It's resistant to a viral disease. And many people believe that even though it's engineered to be resistant to a particular viral disease, it can actually cause more types of viruses in that species. I'll explain why. By the way, I don't ever talk about this stuff. I'm making it up as I'm going along. I mean, I'm making it up. I'm like drawing it from all these different things. You know, I have the whole prepared thing, which we'll go through in rapid fire in a moment. But it's just exciting for me to get a chance to speak about the environmental impacts, which I usually just do, as I say, in the Q&A. So viruses, viral genes, they create viral proteins. Now, viral proteins help the viruses by disabling an organism's defense against viral infection. So you put out crops where you have viral genes in every single cell, creating viral proteins in every single cell, and what does it do? Well, the specific viral gene is correlating with a specific virus that it's defending against. So the papaya defends against the ring spot virus. So you won't get the ring spot virus in the papaya. But now the papaya has all these viral proteins in it which disable the viral defenses against other viruses. And so it could potentially create a problem there. There's a number of other reasons why, and rather technical reasons, why the crops are more at risk from viruses due to the insertion of a viral gene. And I won't go into those details because there's other stuff I want to talk about. Another piece is that, so we talked about two things, collateral damage in the DNA and the actual trait causing problems. And we can theoretically surmise that if you end up with salt-tolerant crops or drought-tolerant crops that are artificially created and not through the process of natural selection, and you introduce them into an area for agronomic impact, you can end up causing a push-out of other species, an invasive type reaction, especially when they cross-pollinate. We'll talk about that in just a minute because that's one of the most serious environmental concerns about GMOs. But back to the actual physical creation of the DNA, the transgenic DNA. When you insert the gene, the process of inserting the gene can cause mutations in the transgene. They looked at some corn, and they looked at the transgene. It turns out only 70% of it got integrated. 30% was chopped off. And so it creates a protein that was never intended. In fact, the protein is a combination of genetic material producing the protein from the insert as well as from the plant itself. Others have little tiny mutations inside. Others have scattered several copies inserted and they didn't even realize it. Some have sections or fragments that are inserted. So the actual protein, the amino acid structure that's created from the transgene could be different than what you intend. And if it's exactly the same, it can still act differently. The same amino acid here as over here. This one's natural, this one's transgenic. And this one may be deadly, and this one may be harmless. So what's the difference? Well, I was interviewing a Monsanto former employee recently who had been studying prion disease, mad cow disease and others, in the late 90s when he was selling genetically modified cotton into California for Monsanto. And one of the scientists told him that there was some extra proteins that were not intended in the cotton plant that had come about through the process of genetic insertion. And he was not worried about it. And this other person was shocked. He said, well, aren't you going to test it to see if, at least if it's not a prion, which might be a deadly disease for animals and for humans? And the PhD said, I've never heard of a prion. And then he found out that these cotton plants, which were in the testing stage, were being fed to cows right near his home. So they're being put into the food supply. And prions probably can transfer from meat into humans, causing the human version, which is deadly, with about up to a 30-year incubation period. And he was shocked, and he said, we've got to stop this because these could be prions. The person said, we've always fed the results of our field trials into the food supply, and we're going to do it now too. So he went around to his other teammates on the Monsanto team, and no one would pay attention. In fact, they ostracized him. So he tried to blow the whistle. He went to the ag commissioners. He went to the professors at the University of California. He said their eyes just glazed over. They had no idea about the possible side effects that could be damaging to health or the environment by these misfolded proteins. That's what a prion is. Almost any, I'm told, I'm not an expert at prions, but I'm told with a new discovery that almost all, or maybe all proteins there can be changed into a prion if they're misfolded. So now, how do proteins get folded? In some cases, the gene has a gene product of a protein that's a big long ribbon, and then there's these other chaperone folders that come in the cell and make the change and fold it up. But you're now introducing a protein that has never before been in that cell. So it didn't evolve over the millions of years in that environment. So maybe it gets folded wrong. We don't know. So this could be the transgene that you're trying to create the protein, or maybe some additional proteins that creeped in that you weren't aware of. In addition, proteins often have molecular attachments, like sugar chains, called glycosylation. And glycosylation can turn a harmless protein into a deadly one through allergies. So they took a study of genetically modified peas. They had taken a gene from kidney beans and put it into the peas. They took the protein from the kidney beans, injected into mice, no problem. They took the protein from the peas, injected into mice, inflammation. And they were shocked because it was the same amino acid structure. They looked at it very carefully and they found, the pattern of sugar chain attachments was slightly altered. And for that, they believe, was the cause of turning these peas into potentially deadly allergenic peas for humans, and they never commercialized them. Now, those are three things then. We've looked at collateral damage of the DNA, the protein that's intended, messing things up, the protein that's intended turns out not to be intended at all. We also talked about a fourth, and that's the overuse of herbicides or chemicals. So the bioaccumulation of agricultural chemicals. The last one is gene transfer. Typically, there are barriers to genes transferring between species. Bacteria exchange genes all the time, but we don't know the rate of transfer between, say, a plant that's being broken down in the soil and the soil bacteria. One of the guiding principles of why genes don't transfer so readily between kingdoms is that the structure of the gene in bacteria is different than the structure of the gene in plants or mammals. And typically, for a lot of types of bacteria, you need a similarity called homology. Now, when you put in a transgene from a soil bacteria or the bacteria growing in the dump site behind Monsanto's factory, and you put it into a plant, now you have a similar looking sequence. So you have reduced some of the barriers to gene transfer into bacteria. They took some sugar beets and they cut them up and they put them into soil, and they found that the antibiotic resistant marker gene had now incorporated into soil bacteria. So it's possible that when you plant a field of genetically modified crops and then it is plowed under, you may have a reservoir of those transgenes hanging out. And you don't know what that will mean when there's Roundup-ready traits and genes hanging out in zillions of microorganisms, or BT creating microorganisms, or viral protein creating microorganisms. It's a real crapshoot. And they don't actually have significant testing of microorganisms to find it. In fact, most vast majority of the microorganisms in soil have never been characterized. More than 95% have never been characterized. So it's a very unexplored territory. I just described 5 categories of problems that have impact both on health and the environment. And I'm going to show you a quick slideshow in a few minutes about the health impacts. But there's one aspect that's just environmental, and that's cross-pollination. When you plant a genetically modified field of corn, and it tassels at the same time as the non-GM corn downwind, you're going to get transfer. So the Pandora's box, it's out of the box. The genie is out of the bottle. So now you have transfer of these traits in an unintended way. There is a product called the terminator, which is a gene which causes the crop to become sterile, so you can't replant the beans or the grains that they would like to introduce. So far they haven't. But it's a leaky technology, it's not perfect. So it's possible that it could cross-pollinate with a neighbor's seed, and the neighbor actually saves seed each year, but now a percentage of his seed will die. It will never germinate because it has the terminator. You could probably imagine the different combinations and permutations of transferring unintended genes into other non-GM crops, but also wild relatives. Canola is in the mustard family, and there's wild mustard weeds. So it's now cross-pollinating with weeds. Genetically engineered canola, engineered not to die when sprayed with weed killer. So now you have weeds that have that trait. So those weeds cannot be killed by weed killer. And you have Roundup ready or Roundup resistant crops. You have Liberty Link or Liberty resistant crops, 2 different herbicides. And you even have a non-GM version of another herbicide resistant weed killer or herbicide resistant variety. There are some canolas and weeds that actually have multiple resistance because of cross-pollination. They don't get killed from Roundup, they don't get killed from Liberty, they don't get killed from the third. So you're creating super weeds. But even the canola itself becomes a weed pest. Because let's say you want to grow a non-GM canola in that field. You can't kill last year's volunteer crops. Volunteer crops, how many people know what a volunteer crop is? Volunteer crop means let's say you're doing canola. You harvest the canola, a lot of the seeds fall to the ground, 10% let's say, and are never harvested that year. Those can germinate the next year, or the next year, or the next year, or the next year, most the second year. So they come up. Genetically engineered, herbicide-tolerant canola. But let's say you decided to plant non-GM canola. You can't spray it with Roundup anymore. You have to spray it with a more toxic herbicide. So you end up with, there's articles now where the biggest weed threat now in Western Canada is genetically modified herbicide-tolerant canola. Because it's hard to kill. And that's why it's on the market, because it's hard to kill. And it's not just cross-pollination, there's also blowing in the wind, seed movement. A friend of mine, Percy Schmeisser, has a 2-mile-long truck route along his farm, and people bring the canola to market. The wind blew the canola onto his land, and he had herbicide-tolerant canola growing on his non-GM canola fields, and ruining his 50 years of seed collection and seed saving, and he was sued by Monsanto. Because he had Monsanto's patented, genetically engineered genes growing in his field. And the Canadian judge said, Monsanto's correct. It doesn't matter how those genes got there. It could be through birds or insects or wind or cross-pollination. But because Monsanto has the patent to those genes, they are able to take all of his crops. So that's an example of how cross-pollination affects farmers and seed movement affects farmers. But in terms of the environment, here's another example of seed movement and cross-pollination. Prodigy created a corn that was engineered to produce a pig virus for diarrhea. They just put in a gene to turn the corn into a factory. And they grew it in corn outdoors. How stupid is that? And the next year, some of the volunteer corn grew up in a soy field and was harvested with the soy. It ended up in a silo of 500,000 bushels of soybeans. And it was just about to enter the food supply and it got caught, so Prodigy had to pay $3 million and destroy the soy. When that happened, people became aware that just 2 months earlier they had to destroy 155 acres of corn in Iowa, my state, because another genetically modified pharmaceutical crop probably had contaminated that field. So we have now pharmaceutical crops outdoors. We have herbicide-tolerant, virus-resistant Bt crops outdoors, exchanging properties with wild relatives and other non-GM crops. And how do we clean it up? That's a problem. We have no technology right now to identify from the air, from visual inspection, the differences. There's centers of genetic diversity, like for corn, it's Mexico, where they have 51 different land races of corn varieties. And so those are important because in the case of mass infestations that wipe out certain corn genetics, they can go back and cross from the original and create all sorts of combinations. And so Mexico had a rule, no planting genetically modified corn in this country, because they wanted to protect the genetic purity of the world center of origin for corn. Well, a university professor from Berkeley discovered that there was corn contamination from GM in these very remote regions. Because of NAFTA, U.S. subsidized corn is being produced at rates that are cheaper than the Mexicans can provide, and so it's taken more than 100,000 farmers off the land, and they've increased as like 6 million metric tons of corn have flooded the market in Mexico, and corn farmers sometimes plant that corn. They don't know it's against the law. They try it out, and it gets transferred to the indigenous varieties. So you end up with movement even where you try to isolate an area. It moves. When you talk about trying to isolate an area, think about Hawaii. It was pure lava rock, more than 2,000 miles away from mainland. Now it's a lush tropical paradise. How did it get those seeds? Nature conspires over the long term to spread her bounty. When you're driving around Hawaii, you can see where the lava flows are because, that lava flow was 5 years ago, so there's only a certain sparse variety of plant life. And that one's 15 years ago, and we can see seed movement in there, and 30 years ago, and 100 years ago, and you can see clearly how nature conspires to move seeds and populate areas. We have no technology right now to prevent that. And at the best case scenario, indoors, in greenhouses, it's still not ironclad. I've seen canola seeds that were scraped from the mud of the boot of a farm worker from a field trial canola that was never legally approved, and it was just approved for this field trial. But it got out of there because it was attached to the mud of the boot of one of the workers. And birds can take it, and tornadoes can take it, and floods can take it. We don't have a technology to stop what nature has been designing and doing for thousands of years. And yet, a biotechnology representative said in a committee meeting in the UK years ago that she said, be more likely to get pregnant from a toilet seat than being contaminated from a genetically modified crop. However, in August of this year, the United States Department of Agriculture admitted that Bayer Crop Science had field trials in rice in 2001, and not since then, but somehow it got spread throughout the rice supply of the United States. And within 2 days, the rice futures dropped by $150 million. There's now 25 lawsuits. And what does Bayer Crop Science say? In contrast to the toilet seat analogy, must have been an act of God. So that's my impromptu environmental talk. And it is quite different than what I normally talk about. I'm going to give you a taste of what I normally talk about. And I'm going to do the fastest PowerPoint presentation. Actually, I'm going to give this tonight. Where am I speaking tonight?
In the
South Underground Lecture Hall, the big lecture hall, underground lecture hall on the other side of campus.
Okay, I'm going to be, what I'll do is I'll tell you a couple of stories before I look at this and just pick some fun slides before, because we're running out of time and I want to get some questions. I said in the beginning I would answer questions and I lied, but I will get to that. The reason why this technology is on the market actually is because of lies. The reason why we're all talking about it now, while we're eating it in our food, is because of deception. My book is called Seeds of Deception. And the reason why it's so popular is because it's not a science textbook, it's a storybook about deception, about rigged research, about hijacked regulatory agencies, about scientists who were threatened by a senior Mexican government official who claimed, we know where your children go to school, trying to get them not to publish evidence, about political appointees overruling the scientists at regulatory bodies. I'll give you one of the most important examples. FDA. 1992, they came out with a policy which said the agency is not aware of any information showing that these foods differ in any meaningful or uniform way. And on that basis, they said if Monsanto believes its foods are safe, they can put it on the market without even telling us. No safety studies are required. The environmental studies are basically, I don't know the details of them, what exactly is required, but I have read analyses of them, and they're very thin in what they're looking for. But let's stick with the FDA. On the basis of that one sentence, we have all these foods because they were put on the market without long-term studies, without any required studies. In 1999, a lawsuit against the FDA forced them to turn over 44,000 documents. And in those documents, it became aware that this sentence was a lie. Not only were they aware that there was differences, it was the overwhelming consensus among the scientists at the FDA that genetic engineering is different and leads to different risks. And trying to force it into the concept that it's an extension of natural breeding was inappropriate scientifically. And what did the scientists say? They said it could create allergies, toxins, new diseases, antibiotic-resistant diseases, and nutritional problems. They had urged their superiors to require long-term studies. So why didn't they? The White House had told the FDA a few years earlier to promote biotechnology. And so the FDA created a position, the Deputy Commissioner in charge of policy. And they invited a person, they created a position for this person. This person was Michael Taylor. Michael Taylor was an outside attorney for Monsanto, and also a biotech group of manufacturers. And in this position, he was in charge of the policy. And the rewrites of the policy systematically removed the scientific concern, removed the science at all, so that one scientist in a memo that became public said, what's become of this document? It's a political document, but there's no science in it. And there's no focus on the unintended side effects. Later, that Michael Taylor became vice president for Monsanto. Now, when they submitted the policy, which was very user-friendly, to the White House said, you know, there's 13 pages about the potential environmental risks. That's too many. You need to reduce that, because it gives the wrong impression that these things aren't the same. And so as it went up the political ladder, the technology got safer and safer. But when you read the actual scientific evidence, or the scientific concerns, it's very dangerous. And there's also a lot of rigged research designed to avoid finding problems and a number of other things. I'm now going to show you just a select few of these studies, which I'll be showing tonight. So this is a preview. Bovine growth hormone, we didn't mention, causes over 20 different symptoms to cows, including mastitis and crippling and early death. We're just going to go past it. Milk from the cows has more pus, antibiotics, and hormones. The hormones are related to cancer and fraternal twins. And Michael Taylor was in charge of that as well. There are some whistleblowers. It's a trail of whistleblowers and attempted bribery to get that on the market, which you'll hear about tonight. GM crops, that's the sentence from the FDA. Not aware of any information showing that the foods derived by these methods differ from any other foods. And that is a lie. Now, okay, The rats refused to eat the tomatoes during a test of the genetically modified tomato. Turns out, farmers and scientists and reporters point out that when given a choice, they have found that animals avoid eating genetically engineered crops in many instances, including geese, pigs, cows, squirrels, elk, deer, raccoons, mice, and rats. In this case, 7 of 20 rats developed stomach lesions, 7 of 40 died within 2 weeks. The only human feeding study ever conducted verified that genes from soybeans transferred into human gut bacteria. So we talked about transfer of genes into soil. Imagine the Bt gene transferring to your gut bacteria and producing pesticides inside you for the rest of your life. That is a distinct possibility from eating genetically modified corn chips. In the UK, this man was working on a protocol to develop safety studies. He discovered that rats that were fed genetically modified potatoes developed potentially precancerous cell growth, smaller brains, livers, and testicles, partial atrophy of the liver, damaged immune system. That's a picture of the stomach lining of his rats. The right side is the GM fed, the left side is the non-GM fed. This is the intestinal wall. Right side is the GM fed, the left side is non-GM. So we talked about a lot of this already in the context of environment. This is Bt. So rats that ate Bt corn had all sorts of problems with kidneys and livers and blood cells and blood sugar and kidney weight and more blood cells, and cows died on a German farm, and people breathing in the pollen from a Bt cornfield developed serious allergic type reactions, and pigs became sterile and so did cows when they ate, according to 25 farmers, and people harvesting Bt cotton developed allergic reactions that were similar to those that were sprayed by Bt pesticides in the Northwest. Hundreds of sheep died after grazing on Bt cotton plants. We'll get a chance to look at these tonight, but this is just sort of a preview. Roundup Ready soybeans fed to mice that had problems in the liver cells, the pancreas cells, the testicles, and the offspring. And rabbits fed Roundup Ready soy had enzyme differences. And the offspring of rats fed genetically modified soy flour, more than half died within 3 weeks, compared to 9% of those that were fed non-GM soy flour. And this is a comparison of the size of the rats. The one on the right, the mother ate genetically modified soy, it's 20 days old. The one on the left, the mother ate non-GM soy, it's 19 days old. 36% of the offspring from the GM group were this type of weight. And soon after GM soy was introduced to the UK, soy allergy skyrocketed by 50%. So these are some of the things, these are just a few of the slides that I'll show tonight. Let me just see what else we want to look at. Okay, I'll talk about a deadly epidemic that killed about 100 and caused 5,000 to 10,000 to fall sick, based on a genetically engineered food supplement called L-tryptophan, and how to avoid eating GMOs. This, by the way, is the current amount of crops that are produced in the United States that are genetically engineered, according to USDA statistics. Since you're a class, you're supposed to know numbers, so I'll leave that up for a minute. So that means basically if something is grown in the United States and it has soy, corn, cotton, or canola oil in it, or soy and corn derivatives, and it doesn't say non-GMO and it doesn't say organic, it will be GM. There's also Hawaiian papaya, zucchini and crooked neck squash, Roundup Ready alfalfa, and Quest brand tobacco. How do we stop the genetic engineering of the food supply? Come tonight to find out. I predict it'll be stopped, or I predict it'll collapse within 2 years, and I'll explain why and how we're doing that, and how you can participate this evening. So, but I don't want to get into all that because we don't have enough time to get into all the questions that I intended. So I want to see a show of hands, how many people have questions, and I'll make sure we get to everyone. I'm putting up two questions?
No.
Okay, 1, 2, 3. That's easy. I can go back to the slides. All right, we'll start over here.
I was just a little bit confused by the herbicide-resistant crops. Why would, I mean, you kind of explained, but why would that cause farmers to have to use more herbicides? I don't quite get that.
Okay. In the beginning, in the first 2 years of use, herbicides were reduced in usage. And over the next 3 years, it was mixed between crops. And then over the next 4 years, there was a big increase. And a major contributor to that is that because they were using Roundup herbicide a lot exclusively as their herbicide, the weeds became resistant to herbicide. So now there's pigweed and horseweed and other weeds that now are not killable by Roundup. And so that's one of the things. Secondly, this you don't hear in the textbooks, but you hear from farmers. A farmer is growing soybeans conventionally, and he looks at his neighbor's field, and it's Roundup ready. And he looks at the rows, and it's completely clean. And he looks at his own rows, and there's weeds. And he feels jealous. And there's a sense that you're a better farmer if you have less weeds. And so that's one of the reasons why some farmers told me that they shifted or that their friends shifted. And so getting rid of all the biodiversity turns out to be a sought-after benefit for farmers. And also, glyphosate became cheaper, and it became off-patent, and so that was another contributing factor.
I think another is that farmers used to only be able to apply before the crop came up,
Right.
apply the herbicide. Well, now you can apply after your crop comes up. You can just apply it as many times as you want.
Right, it's called post-emergence. So that's the reason, the primary reasons. Charles Benbrook has this major study on that. Yes?
How do we, as college students, avoid GM foods? Are they only marked non-GMO and everything else that is not marked non, is it all GM?
Yeah. I'll answer that more in a
For
moment.
those crops, not
Yeah,
necessarily
right.
for all.
Oh, but,
Just for these.
okay.
I'll answer more of that in just a moment. There was a question over here. I want to spend more time on that. One more question?
Well, I'm researching the National Uniformity for Food Act in another class, and I was just curious what you think. It's really hard to find information that says the biotech industry is connected to that, but one of the major things that law will affect is GM foods not be labeled.
Right.
How do you feel about that connection?
Well, this is an example of how industry has controlled and dominated the policymakers. The Food Uniformity Act is basically that states will not be allowed to require labels that are not required by the federal government. So, for example, Alaska has a rule that if genetically engineered salmon is produced, it must be labeled as GE in the state. By the way, that's an interesting environmental aspect, genetically engineered salmon. Imagine trying to do a recall from the ocean. There was a genetically modified fish that had a very slight difference in its, I'll get back to your question, in its fertility rate. And so someone did a computer comparison. What if 60 fish were introduced into a local population of 40,000 fish? And the fish grew faster, so the males grew faster, so the females wanted to mate with them more than they wanted to mate with the non-GM fish. And they calculated, and they were shocked, that there was a complete extinction in 40 generations, according to the computer model, based on these two differences in the expression of traits. So this is an example of how little tiny changes can have big effects over the long term. And so they took these genetically modified salmon, the actual salmon that are genetically engineered to grow 4 times faster, so they immediately grow very quickly, and they want to introduce them into the marketplace. But there was a study in Canada where they put them into tanks, and they were fine, both on their own and with natural salmon, when the food supply was fine. But then they did something interesting. They reduced the amount of food, and then all hell broke loose. All of a sudden, these calm, quiet frankenfish turned on their neighbors, they turned on the other GM fish, they turned on the natural fish, they started cannibalizing and destroying each other, and there was complete extinctions in certain tanks and reductions to like 1 or 2 fish in others, because they changed in their psychology and their behavior, which was a completely unpredicted effect. So, back to your question. In Oregon, they tried to create a labeling law to label genetically engineered foods, but that was defeated because of some interesting public relations and disinformation campaign by the biotech industry. But if you have things like GE labeling in a particular state, this law would eliminate that as a requirement. And it certainly would support the biotech industry, as well as the food industry as a whole, because like in certain areas, in certain states, there's lead alerts. If you have high lead or heavy metals, you have to label it, but this would wipe that out too. In my studies of the FDA, I basically see them as the enforcement wing of corporations and not really protecting the public. And if you look at the details, it's amazing how little they do protect the public. Even the scientists and employees of the FDA, by and large, believe that it's not protecting the public. Even a former commissioner said, what bugs me is that people think the FDA is protecting their health and it's not. It's basically in the pocket of corporations. And this kind of legislative impact also was in the case where local counties were creating GM-free zones, like in California. And so the biotech industry rushed into the legislature of all these state legislatures, preemptive legislation which said no jurisdiction other than the state can have any jurisdiction over seeds. So they eliminated the possibility in 14 states so far that any city or county can create GM-free zones. So this is part of the ways of corporate takeover, and which is one of the reasons why in the work that I'm doing, I'm going not after legislation, but after consumer shifting buying patterns and causing a shift in a tipping point so that we'll see the same kind of landslide change that we saw in Europe. So in the last 5 minutes, I'm going to talk about answer your question about what you can do to avoid eating GM and a little bit about what you may want to do to help rid the world of GM. This will be more of the subject tonight.
Can you do that in about 2 or 3 minutes? I just learned that there's an exam scheduled in here at 5 o'clock. That's what all the rumbling is outside here. So if you can do it in about 2 or 3 minutes, that will leave us just a couple of minutes for me to ask them a question to write down.
Okay.
And also, I don't know what you...
I'll have to put these outside.
We may move these outside for those who would like to look at the books.
The last minute was going to tell you some of these. This is a Seeds of Deception, which is my book. This is 2 DVDs and it's audio CD, and this is a audio CD that's in there. These cost $1. You can download them off my website also, seedsofdeception.com. And these are normally 18 and 18, but I'll put them on special for students today at 11. Don't tell anyone. And then there's Future Food and some others there. But to answer your question, I would say, if I were a student here right now, I would create a GM-free school. I would ask the school authorities and the food service to use genetically engineered foods. And the first step may be to label them. And the next step may be to educate students about the potential health risks. And the third step would be probably a natural reduction of the GM-laden products because of consumers changing their buying habits. We have a GM-free school campaign at my institute. I have a director of the campaign. We have Hidden Dangers in Kids' Meals as a DVD in the GMO trilogy. It talks about the health risks, especially to children, but they apply to adults also. And we have a whole way that we can support you. If you want to avoid GM personally, you have to either buy organic or buy non-GM, or look at the label and if there's soy, corn, cotton, or canola derivatives in it, and it's made in the US, it's GM. If it's papaya from Hawaii, it's GM, likely. If it's zucchini or crookneck squash, probably not, but it might be. And those are the only ones, the only crops that you'll be eating. There's also milk from cows treated with RBGH. And so tonight, the impact of what I've talked about environment, they may be like, well, maybe I'll change it, maybe I won't. Tonight, if you come and you look at the details, you'll notice there'll be a huge shift in what people are willing to be put in their mouth from the time we start to the time we end. So I hope that helped in your understanding of some of the potential dangers of genetically engineered foods on the environment. I give it over to you.